Assalamualaikum. I'm going to present about environmental project 1, E1. I'm from group 55, which are group members Syaiful Hamid, Nur Farina, Sasi Kumar, Nurul Fatin, and me myself, Muhammad Tamizi. So, for BOD, biochemical oxygen demand is the amount of oxygen consumed by bacteria and other microorganisms while they decompose organic matter under aerobic and condition, and a measure of the amount of oxygen required to remove waste organic matter from water in the process of decomposition by aerobic bacteria. And for total suspended solids, TSS, solid in water that a filter can trap are referred to as TSS. The water sample is put through a pre-weighed filter in order to test TSS. And lastly, alkalinity. The alkalinity of natural water are alkaline, mostly due to the salt of weak acid. However, weak or strong base may also contribute. Additionally, it serves as a gauge for how well a treatment can counteract a potent acid.
Okay, Assalamualaikum. My name is Chef Fahmi. So, I'm here to explain about the objective for the experiment E1. Basically, experiment 1 is to gain the knowledge of the method used to measure the pH and also the alkalinity. And then next is to be able to calibrate the pH meter. And lastly, to determine the alkalinity of water samples and express this in terms of the SI unit of ppm and also CO3.
The apparatus that have been used in these experiments are BOD incubator, glass top BOD bottles, calibrate DO probe, measuring cylinder, conical flask, pipette, urine and wash bottle. For TSS experiment apparatus that have been used are glass microfiber, disposal aluminium, suction flask 100 ml, 47 glass microanalysis, drying oven, desiccator, and also the distilled water. For the alkalinity test, the apparatus that have been used in this experiment are 25 or 50 ml burette graduated in 0.1 ml, burette support 100 ml graduated cylinder, rubber tips stirring rod, and white porcelain evaporating dish.
Procedure TSS, preparation of the glass fabric filter is inside the fill piece onto the base and the clamp on the furnace. While vacuum is applied, wash the disc with 3 successive 20 ml distilled water. Remove all the traces of the water by continuing the apply vacuum after the water has passed through. Remove the furnace from the base and place filter in the aluminum disc and then dry main over at 103 degrees Celsius to 105 degrees Celsius for 1 hour. When needed, remove this from the oven, disk and weigh. Again, select the sample volume maximum for 200 ml that will yield not more than 200 mg for the total suspended solid. Third, place the filter on the base clamp on the furnace and apply the vacuum where the filter with a small volume of the distilled water to seal the filter against the next. Shake the sample vigorously and continuously. Transfer 100 ml for sample of the filter. Use a large orifice volumetric purpose. Remove of the trace of the water by continue to apply vacuum after the sample has passed through. Next step, rise the pipette and fill on to the filter with small volume of the distilled water. Remove all the trace of the water by continuing the apply vacuum of the water has passed through. And then lastly, carefully remove the disc filter from the base. Dry for at least 1 hour at 103 degrees Celsius to 105 degrees Celsius. Put in the desiccator and
alkalinity test. First, prepare the 100 ml sample in the beaker. Secondly, dip the pH electrolyte into the beaker. Third step is stir the sample with magnetic stirrer. For this, press the measure at pH meter and wait until the reading is stabilized. The next is record the reading into the lab sheet. Following is add 0.2 ml hydrochloric acid into the sample beaker and read the pH reading meter. Finally, repeat the step 6 until certain time and record in the lab sheet.